Hello there. My name is Brian Clarke. I'm a postdoc in the lab of Oliver Stegler at the German Cancer Research Center, and I'd like to tell you today about DeepRVAT, which is our new method for joint modeling of rare variant genetic effects using deep learning and data-driven burden scores. This is a collaboration with some great other people at the Stegler lab and the Gagnier lab at the Technical University of Munich. And I'd like to especially call out Eva Holtkamp, who did a lot of this work as part of her master's thesis. Let's start by talking about why we might be interested in rare variants. As you can see from this GWAS of height on the right, as the minor allele frequency of a variant decreases, or its rarity increases, the effect on the phenotype can increase quite drastically. But rare variants come with some challenges. Namely, they can be very sparse, so they might be present in only one sample in your data set or just a handful of samples. And they come in great numbers. So in a large whole genome or whole exome sequencing study, you might have tens of millions or hundreds of millions of variants, which leads to a huge multiple testing problem in association studies. Before we go into how to overcome these challenges, let's review the standard GWAS procedure, where you go variant by variant and you test for association with your phenotype, which gives you a p-value that has to be corrected for the multiple tests over a number of variants, and those that survive are considered significantly associated with your phenotype of interest. If you have too many variants to use the standard GWAS procedure, or if your variants are too sparse, not present in enough samples to get good statistics, then the simplest thing you can do is use a burden test, where instead of going variant by variant, you might go gene by gene, and you filter variants for those that are expected to be harmful based on some predicted effects. You count them in your cases and you count them in your controls, and you see if these counts or burdens are significantly associated with the trait. In a variance component test, you have some additional flexibility because you don't have to assume that all of your harmful variants are causal or that all of them have the same direction of effect. So in this case, you assume that you weight your variance according to, say, 1 over the minor allele frequency, and you check whether the distribution of effects of the variance has a non-zero variance. You can also do things like take a hand-designed feature map and map your variance into some feature space, and instead of just weighting the variance, you can use the similarity between them to do your association testing. Now, these standard methods work, but they have some limitations, which is why we wanted to develop DeepRVAT. The first is that you can get very rich annotations from deep learning models or from models that predict the expected consequence of a variant. But in something like a burden test, these are really only used for filtering, whereas we want to utilize the full richness of these highly dimensional annotations. In both kinds of tests, you have strong filtering for harmful or deleterious variants, whereas we managed to leverage signal from high and moderate impact variants. Both these types of models assume additive effects of variance, and we are using deep neural networks to learn optimal nonlinear feature maps. Particularly, variance component tests can be computationally inefficient because you're required to invert large matrices in order to fit them. DeepRVAT is much more scalable and can even leverage GPUs for fast computation. And finally, neither of these tests share information across genes. Testing is really done gene by gene. And DeepRVAT takes some steps towards learning from signal on multiple genes. So let's talk about how the method works. There are two phases. The first phase is training. And in this phase, we restrict ourselves to a small number of seed genes. These are genes that have a known association to the trait. And we do this in order to reduce the noise during training, which you might have if you're training on all, roughly 20,000 protein coding genes in the human genome. We annotate the variants in these seed genes, and we pass these vectors of annotations into a so-called gene impairment scoring model. And we get a gene impairment score for each seed gene and each sample. We linearly combine these scores across the seed genes to get a prediction of the phenotype for each sample. And by minimizing a loss function, we can backpropagate the loss through these two models and learn optimal models. The next step, or the next phase, is association. And in this phase, we are really looking at all genes of interest. So, for example, all protein coding genes. And for these, we compute gene impairment scores for each protein coding gene in each sample using the pre-trained scoring model from the previous phase. So now its weights have been frozen. And then we can use these scores and go gene by gene and do standard association testing with our phenotype. You can use any differentiable model for this gene impairment scoring model. We settled on a model based on deep set networks. So we look at the set of variants and their annotation vectors. We pass them through a variant embedding module, which is shared across all variants and computes a latent representation, which is combined in a symmetric function to get a gene embedding, which is passed through a second model to compute the gene impairment score. This has some nice properties. First of all, we can handle any size of variant sets, which will, of course, differ. The size will differ across samples, so this is desirable. It's also invariant to permutations, so we are really considering variants to be unordered in this case, and we don't care in what order we feed them into the model. We can get away with a pretty small architecture and still get good results. The first module has just 2 layers that are 20 neurons wide, and the second module has 2 hidden layers with 10 neurons. We get significantly increased yield in across a number of phenotypes that we took from the UK Biobank study, a large population scale study from the United Kingdom. Here we have 134,000 samples with whole exome sequencing and various phenotypes, and we see that on 4 out of 5 phenotypes, DeepRVAT manages to significantly increase the number of discoveries against a standard burden testing method on a comparable number of samples. Of course, none of this is meaningful if the p-values are systematically inflated. So we check that these p-values are calibrated. And we have used simulations to verify that the false discovery rate is controlled at the level that we expect. We also have checked that our discoveries are biologically plausible. So if you look at DeepRVAT in comparison to various studies on UK Biobank whole exome sequencing data with larger sample sizes, we see that DeepRVAT has higher power to make more discoveries. But also we see that as the number of samples in the study increases, the number of genes that were discovered by DeepRVAT that are then replicated within these studies increases. And we expect that if the sample size is increased even further, then this curve would continue to show that even greater replication of DeepRVAT results. Now, I don't like cherry picking, so I'm just going to show you another phenotype where DeepRVAT doesn't compare quite as favorably to these studies. However, you do see that at a smaller sample size, DeepRVAT still manages to make a higher number of discoveries, but it's not magic. If you increase your sample size by 2 or 3 times, then you might on certain phenotypes make more discoveries than DeepRVAT. Another thing we checked regarding the biological plausibility of our results is a gene set enrichment analysis from the Enricher tool. And we see that DeepRVAT discoveries are enriched for known effector genes of related phenotypes. And here again is the example of HDL cholesterol. One thing that's a little bit preliminary, you might ask, is it really important during the training and association phase to use the same phenotype? And what we found is that in fact, this is the case. So if we look across the 5 different phenotypes that we studied, and we compare using the same phenotype during the training phase and the association phase with randomly selecting the phenotype during the training phase, and then testing for here triglycerides during the association phase, this performs worse than using the matched models across both phases. And this hints that there is something really trait-specific going on in the effect of different variants, which is different from how burden tests and variance component tests are currently done. So to summarize, DeepRVAT is the first method to use deep neural networks to learn nonlinear effects from annotated rare variants in rare variant association studies. This greatly increases the power to discover gene trait associations. DeepRVAT is well calibrated and its discoveries are indeed biologically plausible. And finally, we have some hints that variant effects can be trait specific, again, in contrast to the usual assumptions in burden and variance component tests. Now, believe it or not, in 10 minutes, I could not tell you everything we've done in the last few months. So if you're interested in learning more about this method, please do find us at ISMB if you're going to be there. We will be there in person. or send us an email. And thank you very much for your interest and attention.